Cure Talks. I'm Priya Menon, and today we are discussing messenger RNA COVID vaccines. Joining me on the Cure Talks panel is David Stanley, Matt Goldman, and Dr. Herb Geller. Our featured guest for today is Dr. Drew Weissman, professor at the University of Pennsylvania. Dr. Weissman co-developed the messenger RNA technology being used in the COVID-19 vaccines produced by Pfizer and Moderna. Dr. Weissman, welcome to Cure Talks, and thanks for joining and taking the time to speak with us today.
Thank you.
I think we can begin with a brief history of this type of vaccines, that is the mRNA vaccine now being used in the Pfizer and the Moderna, and you've been working on this for quite a few years. So it'd be great if you can give us a little bit of history of how this has come about.
Sure. So I came to Penn 23 years ago and met Katie Carico at a copy machine. And I studied dendritic cells and vaccines, and she studied mRNA, but wasn't getting very far. And nobody in the RNA field was advancing. So we started to investigate RNA in the immune system. And we figured out that RNA was highly inflammatory. And that was problematic because if you give RNA to a patient, you don't want them to get sick from the RNA. So we figured out what made it so inflammatory, and then we figured out how to avoid that inflammation. And that was our breakthrough in 2005. What that is it's called nucleoside modified mRNA. And what we essentially did is we changed the RNA so that the immune system, our bodies, didn't recognize it as foreign. And that's the RNA that's used in the Moderna and Pfizer BioNTech vaccines.
Mm-hmm. So what you're saying is that the initial issue was that these vaccines, that's the one with mRNA, was highly inflammatory, but with a small tweak, you were able to incite the right amount of immune response and protein production.
Exactly. And we actually got rid of all of the immune recognition, and that greatly increased the amount of protein made.
Mm-hmm. So do you think, do these mRNA vaccines have an advantage over the other type of vaccines? And if so, what are some of these that we are seeing for COVID-19?
So the two RNA vaccines, the Moderna, BioNTech, Pfizer, are the first ones licensed. So this is the first comparison that we've got. And the comparison is 6 months old. So it's hard to say, are they better? Do they have advantages? What we do know is one of their advantages is that they're incredibly quick to make. So if you have to make an inactivated virus vaccine, that's a lot of work. You have to figure out how to grow the virus, how to inactivate it, how to purify it, how to make sure it's safe. With RNA, you only need the sequence of the protein of interest. So for coronavirus, that's the spike protein. And we've known for over 20 years that the spike protein is the principal vaccine component. So the day that the sequence was released on January 12th, we made an RNA vaccine for it. So it's very quick. It's also very effective. We've seen that in the clinical trials and in the patient use, 95% efficacy against any symptoms and 100% efficacy against serious symptoms and death. There are a lot of unknowns still, and we'll learn those over time.
Mm-hmm. Dr. Weissman, so with the new COVID variants that we are seeing, which are emerging across the world, how effective do you think these mRNA vaccines would be?
Well, what we know so far is that the RNA vaccines still appear to be effective against all of the variants so far. What we don't know is what's going to show up in the future. More variants are going to occur. Until we can vaccinate the entire world, variants are going to keep appearing. So the concern is that in the future, a variant might appear that the vaccine is completely useless against. For an RNA vaccine, it's very simple to make an update, a booster, an improvement. I was talking with Ugur Sahin, who runs BioNTech, who said it would take them 6 weeks to have a new vaccine in patients' arms. So RNA allows you to very quickly update vaccines, which I suspect will be important as new variants appear.
Dr. Weissman, the panel has great questions lined up for you, so I'm going to let them start with their questions. David, so we have David here, David Stanley. David is a melanoma survivor, advocate, and author. David, please ask your questions.
All right. So, Dr. Weissman, I do familiar with Dr. Neubig's work at MSU on mRNA interruption, and I know you guys. It's very different the way these treatments, these interventions work. But do you think mRNA manipulation is a holy grail for other diseases as well?
You know, I think it's too early to say that, and I think you're asking a father to, whether they like their children,
right?
So I think the RNA therapeutic platform has enormous potential. It's worked unbelievably well for COVID in 2 different companies' hands. We're doing clinical trials for 5 more pathogens right now. BioNTech and Moderna are doing additional clinical trials. RNA vaccines are in phase 2 clinical trials for cancer as personalized vaccines and have really shown great success or better success so far. RNA therapeutics, monoclonal antibodies are being delivered with RNA. So it has enormous potential. And my guess is that it's going to be, a turning point in medicine. But, we'll have to see.
Okay. When I mentioned to a friend who does virology that I was going to be speaking with you, they shared this study, I think it was Canadian, that came out in January about blood type, and it was correlated with susceptibility and morbidity and mortality. What's your take on that? Is that study something that's worthwhile and should be pursued? Or is this just something that, it's another case of correlation is not causation?
Yeah, so what we don't know yet. I mean, it's pretty clear that certain blood types, A in particular, has increased risk from infection. We see this with a lot of diseases where there are genetic associations with disease acquisition and severity. So I take it very seriously, and I think it deserves more research to understand exactly how that happens.
Okay. Now, we were talking a little bit about variants of COVID-19 popping up pretty regularly because, of course, evolution likes that sort of thing. Do you feel that the vaccine is going to be pretty adaptable? Because sooner or later, one of those variants obviously is going to pop up that the vaccine that we're currently using isn't going to be effective against, right?
Yeah, so there's a lot of variants that keep appearing. They're going to appear until we can vaccinate the world. The only way to stop the variants is to stop the virus. So far, the main variants that vaccines work less well against are the South Africa, the 135. Although the Brazilians are likely to fall in that category as well. And new ones keep appearing. From what I've seen of the data, the vaccines, the RNA vaccines, are still effective against the South African and Brazilian variants. But you're correct. At some point, variants are going to appear. It's going to be like influenza, where variants keep appearing that are resistant to vaccines. We're going to have to either figure out better vaccines for the long term. So what Moderna and BioNTech are doing right now is they're making a booster with the South African variant. And that's great. It'll take care of the South African variant, but it probably won't take care of the next ones that appear. What we're working on is making a booster, a new vaccine that takes care of all of the variants that have appeared and any variants that might appear in the future. And I think that's a better approach. The Moderna approach to making a South African variant is quick. The FDA already said they won't require efficacy trials. Our vaccine is going to require efficacy trials. But if this, it's going to be years before the world is vaccinated. So I think we have to think in the future on how to prevent all variants to stop this.
I have two more questions. They're both, I think, pretty quick. Before you came on, we were talking about our second dose side effects. I felt a little lousy. A couple friends of mine felt terrible. My wife felt, nothing. Dr. Geller's wife was very sick. Is there any data that relates these second dose side effects with manufacturer, or is this just a case of immune systems are funny and they're individual?
So if you look at the side effects, 80 to 90% of people get sore arms, swelling at the site, and a smaller percentage get systemic effects. This is telling us that the vaccines are working. This is the immune system responding to the vaccine. It has nothing to do with manufacturing. It has nothing to do with contaminants or any other problems with the vaccine. That they appear to be purely our immune system responding. So in my mind, it's a good thing if you have an adverse event, because it means the vaccine is doing its job. Not to say that we are working on newer vaccines that have fewer of these adverse events so that the vaccine is better tolerated.
Got it. Thank you for clearing that up too, because there's a lot of talk out there, obviously, about, that this is, the conspiracy theories are wild. This is my last question, Dr. Weissman. I have some familial arthritis in my CMC joints, right at the base of my thumb. Thanks, Dad. And I seem to have gotten, like, within 12 hours of my second shot, I really got some severe arthritic flare-ups. Is this the sort of thing that you might expect when my immune system gets amped up from that second shot?
Yeah, we just don't know. I mean, the vaccines in the phase 3 clinical trial, people with arthritis and autoimmune diseases weren't included in those trials. So we don't know. There's no scientific evidence from any animal studies or earlier human trials that the vaccines will cause a flare of an autoimmune disease. But that's not to say it may not happen. When your immune system is amped up, is boosted by a vaccine, there's a potential that it will recognize other antigens, autoantigens, that it regularly recognizes. So this is something that needs to be studied.
Yeah, because I feel fine. I felt fine. Really, yesterday was the first day. My second booster was on Tuesday night, and my thumbs were fine again yesterday. So 3-4 days, of some arthritis pain is worth it. But I wondered if there were any other kind of implications. Exactly what you went to with the autoimmune discussion. Priya, those are my questions. Thank you, Dr. Weissman.
Also to note that some of the adverse events from the vaccine are arthritic type of complaints. So this could be completely unrelated to your ongoing arthritis, may simply be another adverse event of the vaccine.
Got it. Thank you, sir.
Thank you, David. Next on the panel, we have Matt. Matt, please go ahead and ask your questions.
Okay. First of all, Doctor, thanks for your work and thanks for your time. As far as we know at this point, are mRNA vaccines less effective on cancer patients, or even more specifically blood cancer patients? I'm hearing a lot of both sides of the coin on this.
So there's a big clinical trial of a personalized RNA vaccine ongoing across the world. This came out of earlier work that was melanoma work, where they made personalized RNA cancer vaccines for patients who had an expected survival of approaching 0. And the results of those trials were, I think it was around 25 to 30% achieved remission, some complete remission, which to me is a fantastic improvement. It's not as good as CAR Ts with liquid cancers, but for a vaccine against a disease that has no hope of cure, to get 25 to 30% clinical remission is fantastic. We're still early in RNA cancer vaccines. We're still learning how to make them best, how to choose the right immunogens, how to deliver them, what to deliver them with, checkpoint inhibitors, et cetera. So that, to me, that's a pretty good start. And I think we're going to be learning more and improving cancer vaccines in the future.
Mm-hmm. But in terms of the effectiveness of the coronavirus vaccine on cancer patients and blood cancer patients, is there less effectiveness for them with the vaccines that you've been working on?
Certainly for anybody with an impaired immune system, a vaccine is not going to work as well. So anybody that's had their B cells depleted with CD19 antibodies or CD19 CAR Ts, they're not expected to make much, if any, of an antibody response. They'll still make some T cell response, which offers some protection. We don't have good data yet, but the more immunosuppressed a person is, the less well they're going to respond to a vaccine. And there's nothing we can do about that, because when your immune system is impaired, when it doesn't work well, you just don't respond well to vaccines.
But folks should still get vaccinated nonetheless, correct?
Yeah, even the worst vaccines in clinical trials still protect people from serious disease and death. So any response you get from a vaccine, I think, will help you.
And is it the genetics of so for instance, I'm a multiple myeloma patient. Is it the genetics of the cancer, or I'm on an immunotherapy now, which a lot of people are, myeloma patients, it's really effective. But is it the workings of the treatment we're on, or is it the cancer itself that's sort of changing our immune system and making us less viable, I guess, patients for the coronavirus vaccine to be effective? If that makes sense.
Yeah, I mean, what we know is that multiple myeloma, CLL, other liquid tumors impair the immune system. They impair the bone marrow. They impair immune responsiveness. And that's the effect. So if you've got somebody with CLL or AML that's been cured, their immune system will work nearly as well as a normal individual. If you've got somebody with active disease, their immune system is impaired.
So for vaccinated cancer patients, a lot of folks that have normal immune systems are getting vaccinated and they're feeling like they can go back to living a somewhat normal life. But for cancer patients and immune compromised folks, are they waiting for a certain percent of the population to be vaccinated or herd immunity? What are they looking at in terms of a return to normalcy?
Yeah, so that's a hard thing to address. The first is that we don't know how good a single patient is going to respond to the vaccine. There's probably a lot of variables, and somebody with multiple myeloma on immunotherapy that has a good response they're probably going to respond better to somebody who has much worse disease. So for an individual patient, we don't know how well they're going to respond. And how well they respond is going to correlate with whether they'll get the disease, what the efficacy of the vaccine is. So, on a population basis, we know that people with active cancer have reduced immune responses. But for each individual patient, we just don't know what that means. Ideally, we want to reach herd immunity in the United States and the world to slow this and to stop this pandemic. Once that occurs, people can go back to normal. My guess is that, and I tell this to my immune deficiency patients as well, you need to really wait for herd immunity to be achieved before you can go out without a mask and return to your normal life. I don't know how well those patients are responding to the vaccine, but I say that out of caution because I don't have the answers.
Okay. And sort of related to something that was mentioned earlier, should a patient, is it correct or incorrect for a patient to infer the efficacy of their vaccine if they have zero response to the first or second shot?
We've seen no correlation between adverse events and how well you respond. So it's not that people that have no response didn't respond to the vaccine. They were lucky enough not to have adverse events. No data has been released that associates with that.
Okay. That's all I have. I definitely appreciate your time. Thank you.
Thank you.
Matt. Thank you, Matt. Next we have Herb here, who's a prostate cancer survivor. Herb, please ask your questions.
Right. Thanks for joining us, Dr. Weissman. My first question would be, what do we know about the duration of action of the immune response? I mean, we're only 6 months into this, so we really don't have any long-term data.
No, you're exactly right. And you actually have to look at it two ways. So right now, Moderna, Pfizer, BioNTech are measuring antibody levels over time. And the latest data from Pfizer shows that at 6 months, people are well protected. Antibody levels are high. We can predict that means that at a year, they'll probably be well protected, but we don't know, is the vaccine going to last a year, 5 years, 10 years? Right. The other issue is that a vaccine does two things. It makes memory and it makes effectors, so active antibodies and T cells. They're only measuring active antibodies in the blood. There are memory B cells that respond very quickly. And for all we know, you could have 0 antibodies in your blood, but if you've got good memory cells, you'll make a response fast enough where you're completely protected. So it's really going to be following people over time and seeing when do they start getting disease again. And that will tell us when the vaccine needs to be boosted.
Right. So yeah, I mean, the other question is related, and that is all these vaccines are all against the spike protein. And when you're using mRNA, you're using a discrete region of the spike protein versus the traditional method where you're using potentially a larger segment. So are there regions of the spike protein, for instance, which are less subject to mutation that would be better targets, or do they happen to be the ones that are not immunogenic?
Yeah, that's a great question. So another thing that my lab is doing is we've been working on a pan-coronavirus vaccine. And what that means is that there have been 3 coronavirus epidemics in the past 20 years. It would be foolish to not think that we're going to have more. We're definitely going to have more coronavirus epidemics and potentially pandemics in the future. So what we started doing last summer is trying to make a vaccine that would prevent or protect against any bat coronaviruses that have the potential to infect humans. The way we're doing that is we're looking at conserved regions of the entire virus, as well as conserved regions of the spike protein. And we're trying to make vaccines that induce those responses. We've had some success. We've made a vaccine that can prevent SARS, which was the first coronavirus we know about, and COVID-19, as well as all of the bat coronaviruses we've tested. So it looks like we have the potential to make a pan-coronavirus vaccine. But you're right, it's identifying the right immunogens, the right regions of spike and the entire virus to use in a vaccine.
So getting back to a continuation of one of the previous questions about cancer vaccines, I mean, mRNA can be essentially ordered with any sequence you want. And so, I mean, and then there are lots of potential antigens on the surface of various cancer cells. However, I mean, liquid tumors seem to be much more susceptible to immunotherapy than solid tumors in terms of just in general. So do you think mRNA techniques can overcome that, or you think we still have to deal with other issues like exhaustion and things like that?
Yep, so the clinical trials that are ongoing are looking at all of those issues. The first one was melanoma, which is a solid tumor
Right.
that responded well. The clinical trial includes many different types of cancer, including breast, colon, lung, and renal, and others. Part of the trick is that tumor cells are our cells that have mutated. So identifying the right antigens that will kill the tumor cell, but not kill our own good cells is a problem. So that's being investigated. The vaccines are being combined with what are called checkpoint inhibitors, which are they're antibodies against antigens that tell a cell to become senescent, to stop working. And by blocking it, you wake up those cells. So it's a multifactorial approach to making a good cancer vaccine. And they're in clinical trials, they're showing effectiveness, they're being improved. I have great hope for the future.
So in general, I mean, vaccines are one way, but there's actually lots of different immunotherapy approaches these days in immunotherapy of cancer. I mean, CAR T cells, bispecific antibodies. Do you have any sense of which are the most likely to be successful? You think they're all eventually going to be figured out?
Yeah, that's a very complicated question, and I don't have time to answer it fully. But there are many immunotherapies. The CAR Ts against liquid tumors are fantastic. They have not worked well against solid tumors.
No.
We're trying to figure out why that is and ways of making them work better. So my guess is that we're going to need many different immunotherapies, CAR Ts, BiTEs, cytokines, and vaccines to interrogate all of the different types of cancers and see which ones work best for each type of tumor.
So, and then one last question for me for now. I mean, getting back to the COVID situation, is there a difference in the antibody repertoire between mRNA-based vaccines and attenuated virus vaccines?
So there are a couple of differences, and this hasn't been investigated well yet. We've investigated it in animal models. What we see is that, the RNA vaccines give much higher levels. So even in the phase 3 trials, the level of antibodies in vaccinated people were about 5 times higher than convalescent patients. Other vaccines, the adenos, typically give levels similar to the convalescent patients. So the RNA vaccines make higher levels of antibodies. As part of that, some of those antibodies neutralize the virus, kill the virus before it can infect the cell. Those levels are also higher. We're looking at specificities, so particular parts of the spike that the vaccines can recognize. And we've identified some that RNA induces well, that infection doesn't induce. And some of those are conserved antigens that may offer broad protection. So those are new and ongoing studies. In general, the RNA vaccines are better than most other vaccines. They give higher levels of antibodies.
Okay, well, thank you for now. I'll let my co-panelists continue.
Thank you, Herb.
Thank you.
Dr. Weissman, I had one more last question before, I just want to circle back before the internet cut me out. So I know that, like, I think you just touched upon how mRNA incites a response, but my question is, why do we need 2 shots to make mRNA vaccines create more antibodies, or why is there a more pronounced reaction after 2 shots? And what is that shows about delaying the booster dose?
Yeah, so those are great questions that we don't have answers to all of them.
Mm-hmm.
We've developed probably over 30 different vaccines using mRNA for everything from Zika to Ebola to genital herpes, HIV, influenza, hepatitis C. Some of those vaccines work well with a single injection. Others require multiple. If you look at the phase 3 trials, both the Moderna and the Pfizer vaccines are 80% effective after a single immunization. What we don't know, and what the concern was, that how durable that response is. And that gets into basic immunology. And in basic immunology, the first time you see a pathogen or an antigen, the response is limited, and it's usually not very potent. And the immune system requires a boost, a second vision of that pathogen to make a better response. And that's why we give most vaccines are given 2, 3, 4 times to boost and to improve the response. We see the same thing with RNA. The titers, the level of antibodies, goes up about 10 to 20 fold when somebody gets a booster. And that's why you go from 80 to 95% protection. The effect on longevity we're now investigating. Nobody or very few people got a single vaccine with the RNA. So it's going to be hard to know, will one vaccine dose give you years of protection? But people wanted 95% protection and good durability, and that's why they get 2 doses.
Thank you, Dr. Weissman. I think we have a couple of more questions from the panel in 5 minutes. So David, you can go first, and then Matt will have yours.
Okay, Dr. Weissman, as a high school science teacher, I can tell you that not everybody is as familiar as those of us on the panel are as lay people. Well, Dr. Geller's not a layperson, but could you give us a quick rundown? We've been throwing around these words, mRNA, and could you give us a quick rundown exactly what is the role of mRNA, DNA, kind of a little basic science refresher for people who are listening to this that might not be as familiar with the conversation that we're having?
Sure. So the way a cell and our body works is that our DNA contains all of our genetic information. So every protein in our body that makes up our body is coded for in the DNA. the way the body makes a protein from that DNA is it uses an mRNA, a messenger RNA. So an enzyme copies a protein off of the DNA. The RNA then travels to a machine. That machine is called a ribosome, but it's essentially a machine that can read the code in the RNA. And the RNA vaccine works is that we encode the spike protein from as an RNA to a cell, and the ribosomes, the cell machines, read the RNA and produce the spike protein. The body then recognizes that spike protein as foreign and makes an immune response against it.
That's perfect. I think people will appreciate hearing that.
Thanks, David, for that question. Matt, you can ask yours.
Yeah, thank you. Where can we learn about some of the trials that are going on that you mentioned a little while ago, Doctor?
So the trials that are being planned for haven't been submitted to the clinicaltrials.gov website. That's a website where you can learn about any clinical trial going on in the world. We have published papers in scientific journals that describe these vaccines, but these aren't understandable to a lay audience. So, I think the first time a lay audience will have broad exposure to them is when they're on clinicaltrials.gov and the media starts talking about them.
So we just need to stay tuned at this point.
Yeah, sorry about that.
Okay, no, thank you.
Thank you. Thank you. Thank you. So I think that kind of wraps up all our questions. Dr. Weissman, thank you for your time today. And I want to congratulate you for your instrumental role in bringing this technology of mRNA vaccines for COVID and kind of help to save the world. So it's been a pleasure talking to you. Thank you very much. Matt, David, and Herb, thank you for joining and all the great questions. I hope you put everything that you wanted to. So it was great to have all of you here on Cure Talks. Thank you, everyone.